afternoon, everybody, or good evening, depending on where you are within the world. My name is Bruce Whitelaw. I am the Interim Director at the Roslin Institute of the University of Edinburgh in Scotland, and I'm here to moderate and introduce a 90-minute session, which will start with my colleagues giving short presentations, and then we'll move on to a question and answer session. The whole session title is called Genome Editing, Disease Resistance and Animal Welfare. And my fellow panelists are all involved in the use of this exciting transformative technology. I'll introduce them first. Anna Wargalius, who is out of Norway and at the Institute of Marine Science, Marine Research. My colleague Rachel Hawken, Head of Genetics in a company called Cobb Van Tress, which is one of the leading poultry breeding companies in the world. And Rachel is speaking to us from Arkansas in the US. And lastly, it is Apollinaire Duquesne, who is the director of the Center for Tropical Livestock Genetics and Health, which is a collaboration between the University of Edinburgh and the International Livestock Research Institute in Nairobi in Kenya. Apollinaire may look like he's in the Bahamas, but I can reassure you he is in wet, cold Scotland at the minute, while Anna obviously in Norway and Rachel in Arkansas. This whole session is going to try and introduce briefly the technology of genome editing, followed by some examples and where we are within the livestock arena, the farmed animal arena, and finally raise questions and give you as an audience a chance to pose questions to this panelist. But before that, I want to ask you to participate in a very small poll. We're going to try and do this at the start of this session, but also at the end of the session. So I'm hoping that the screen will be shared with you, which describes this question I want to ask. I'm not sure if I can see this, so can somebody indicate to me if the screen is actually up?
It is up.
It's up. All right, so as you'll see, there's a question there, and it is, do you agree with the use of genome editing to tackle animal diseases? There is also at the top indication of a website. That website is www.menti.com, menti.com. If you go into your smartphone or your computer and type in that website, it will take you to a page which asks you for a code. The code is 9226613. I'll repeat that, 9226613. That will allow you to go in and answer one of apply an answer to this question if you feel that that is what you want to do. So the question is, do you agree on the use of genome editing to tackle animal diseases? And there are three possible answers we've offered you. One is yes, one is no, and one is don't know. If you could please do that, and hopefully we'll see some responses coming up on the screen. I'll repeat that. If you could use a smartphone or a computer to go onto the website www.menti.com and type in, once you go into that website, you'll come to a code which is asked for, and that code is 9226613. Then you'll be given an option of three answers, yes, no, and don't know. And if you could apply what you believe is your feeling about that question.
Okay. Dear all, thank you for invited to have this presentation online, presenting some of the work that we're doing in Atlantic salmon and salmon aquaculture on gene editing. We have not been working too much with disease resistance because we believe that it's better to start in the end where the animal are sterile. So I'm just going to tell you about the project we are working on, which is called the virgin salmon, in which we are then working on production of sterile salmon, which we then can work on editing to introduce disease resistance. So in salmon farming, as you can see, they are farmed in sea cages like this, and they can then escape. And in Norway especially, there is a lot of wild strains of salmon that are then affected by these farmed escaped salmon. So there has been a large demand for having sterile fish. And in this context, we have then developed the sterile salmon by knocking out the gene, which is only found in germ cells of the fish. And then when you remove this gene, both the females and the males become germ cell free and sterile. And in addition to this, the fish are also doing very well in terms that they don't enter maturity, so they don't have the problems with increased disease susceptibility and loss of growth and bad welfare associated with early maturity. Of course, these fish are not too easy to use. And I come back to that. I just wanted to show you how we do this gene editing in salmon with the movie that I have here. These are 4-year-old gene edited salmon that we then mutated to become albino. So they're sort of the first step that we made them 5 years ago. But we're then working with secondary maturity. And these are then the sperm, and these are the salmon eggs, and we mix them to fertilize the eggs. And here we are preparing then the CRISPR experiments, and these eggs are quite big, and there's just a little cell on top, so we have to turn every egg to be able to inject the eggs. So here we have to inject every single egg, and that's a little bit why it's not good. You cannot do it to make all the fish in production sterile by doing this, and that's why I'm coming back to the methodology that we have developed then to be able to produce a lot of sterile fish. Because when they are 4 years old, this is when they breed. So in this project I'm talking about, we then removed one gene and then we also stopped reproduction. But then we developed a rescue technology in which we first knock out this gene, but we also add this gene when we knock it out. So then they still get germ cells, both eggs and sperm. And then you can make new brood stock and add in the next generation, or you can have them produce production fish, which are then germ cell free. This is the sterile fish. And this is just showing an illustration how it looks like. This is what we call the virgin fish, which then have germ cell, although they are genetically sterile. So this is then the first stop for us. Now we want to develop this fish further, and we're waiting for this new generation of this rescued fish, or the virgin fish, in which we then can introduce edits which has to do with disease resistance, because there are some major diseases problem with the salmon in aquaculture. They have this problem called salmon lice. It's a parasite that's killing off the wild salmon, which is the largest problem, but it's also a big welfare problem of the fish in the farms. So we are currently having projects where we look at the proteins involved about the recognition, infection, and egg production of these parasites. So maybe if we can find edits that can then make them resistant to this lice, it would be very beneficial for both the wild fish and the fish in the farm. So that was everything I wanted to say, and thank you for listening.
First of all, my apologies to everyone for disappearing like that. Not my intention, but you may gather that I'm having connectivity issues today. My colleague is kindly showing you the slides that I would talk to, and I want to give you a very brief story about some of the work that's going on at the Roslin Institute at the minute. And this relates to a disease in pigs. Ines, could I have the next slide, please? Pigs, one of the major protein sources in our agricultural livestock system, suffer from a number of diseases, and some of you will be aware of those diseases. One in particular is called Porcelain Reproductive Respiratory Syndrome disease. And unless you are involved in pig farming of one sort or another, you will not have heard of this disease. It only affects pigs. We are very clear about what causes this disease. It's a virus called Porcelain Reproductive Respiratory Syndrome virus. We have no robust way of treating pigs that have this virus. There is a vaccine, but it's not effective, not fully effective. And the result of infection is usually death upon the animals. It is a very severe disease, and probably the number one, or at least one of the major viral diseases in the pork industry worldwide, causing millions and millions of pounds of devastation to the industry, causing an awful lot of distress and harm to the animals, and leading in some cases to farmers losing their occupation and livelihood. Could I have the next slide, please? We know a lot about this virus, and in this slide, there's a protein shown in diagram form, the blue shape of it. It's a modular protein that sticks out of the surface of a cell within the pig. It's called CD163. Now, you and I also have this protein, but we have a different variant of it that this virus does not recognize. Ines, could you press the forward button, and hopefully we'll see a little movement in this virus. The virus moves and binds to part of the protein called domain 5. Now, we know this from extensive work in laboratories and in vitro and cell culture studies. We know precisely which bit of the protein this domain 5. Ines, could you advance the slide again, please? Domain 5 is encoded within the gene that is the gene that encodes for this protein, and it's coded by an exon. It's called exon 7, and we have used the CRISPR technology, these genome editors, that we can direct to specific parts of this gene using guide RNAs, and then using the Cas9 protein, we can cut out, just like a pair of scissors, this exon, and in doing so, we remove the domain 5 from the protein. Ines, could I have the next slide, please? Here we have a diagram showing you CD163 now lacking domain 5. Again, Ines, could you advance the slide? Now that the virus comes along and it does not bind to the protein, it does not bind to this receptor which sticks out of the cell surface. We have generated, next slide please. We have generated populations of pigs, several generations now, which carry this slightly truncated protein called CD165 delta 5, CD163 delta 5. These animals are completely resistant to the virus. Now, we haven't tested large numbers of animals, and we have tested animals specifically in containment facilities rather than in farm or in more real life environments. But in those containment facilities, these animals are entirely resistant to the virus. They show no viremia, they show none of the symptoms, they do not exhibit any presence of the virus post-infection, and they do not show any antibody response to the virus. To all intents and purposes, although the pigs are infected, they do not show any of the symptoms that the virus, the disease, porcine respiratory syndrome, suffers from. Now, obviously, at the minute, these animals are being bred up in a commercial environment to test in much larger numbers whether this resistant holds true. I should also say that we've looked at other properties of the animals. They reproduce as effectively as normal. They are the same size, they grow at the same rate, They show all the other properties of what a non-edited pig would be, except from the fact that they do not suffer from this virus. At Roslin, we think this is a very good example of how this new technology can be used to create disease-resistant animals that can be of use in the livestock arena. And I'll stop my presentation here and ask that we move on to the third presentation, which is by Rachel Hawken. Now move to our third presentation, and we jump species again. We're going to have a presentation from Rachel Hawken, Head of Genetics in the Cobb Van Dresk company. And Rachel's going to tell us about how this technology could be applied within poultry. Rachel, please.
Thanks, Bruce. So I really appreciate the opportunity to present a perspective from the poultry industry in this presentation today. So for the poultry industry, when you look at the number of global supplies of genetics for any of the avian species, there are very few, which means that there are very few companies that are involved in the genetic improvement of that species, improving feed efficiencies, improving welfare, etc., of those livestock species. That means also that those few genetic suppliers of our avian species must provide a product that has an anticipated performance in all global areas, despite global differences. What do I mean by global differences? What I mean by that is that the product or the avian species, the animal, the bird, must be resistant to environmental changes, so heat differences between countries. It must be resistant to management issues, which is what type of feed, what type of housing do the birds have, what type of nutritional differences that are in different global areas, and also cultural differences that we have globally. In addition to that, the avian species must be resistant to multiple pathogens and disease challenges. When you think about a global environment, some countries have different pathogens, and they also have different vaccination schedules, different antimicrobials, different rules around antibiotic use, and of course, different welfare standards. So when you put all this together, the few global genetic suppliers for avian species really have to have a crystal ball. They really have to be able to predict global performance over multiple decades in order to keep affordable protein being produced and available to consumers. So what are the solutions to this? Well, first of all, most genetic improvement companies have a genetic improvement program where they select superior breeding stock based on performance and genetic potential. using just traditional genetic improvement programs, this does not require genetic modification. It just requires information about genetics, information about performance, so that our companies can keep the best birds, breed the best birds together, and produce populations that improve year over year. So it is disease resistance in avian species is actually quite difficult because we can do our genetic improvement in our weight categories, our feed efficiency categories, our welfare categories that we biosecurity. For example, we cannot bring diseases, known diseases, onto our pedigree farms because that will cause problems and we will not be able to supply product to our customers. So while we can make improvements in our traditional performance traits, making improvement for disease resistance is difficult and therefore a priority for when you do an internet search for the cost of avian influenza in different countries, you'll get an enormous range of records, but they're all in the billions of dollars annually loss from avian influenza. And it's not just the loss of birds, it's the loss of product, it's the loss of availability of protein for different countries. So in collaboration with the Roslin Institute, we have set or a presentation recorded from ACO that details what we have done for this effort. This effort is not complete. We have produced birds, but we are not finished with the research and development on this product. The second effort that we have attempted is to create a tool. We call it a surrogate host, to create a tool that is necessary to retrieve a chicken from a cell. So you do a gene edit in a cell, but how are you going to make that cell a chicken? Remember, chickens are not mammals. They all come in the form of an egg, and the egg hatches, and that bird grows. So we have developed this surrogate host development necessary to retrieve chickens from stored cells. Those stored cells can be edited or not edited, depending on the use of this surrogate host. Thirdly to that, there are many opportunities in the area of disease resistance. The question is, what has the biggest global impact? And I'd like to finish by showing you a family picture of some of our surrogate host work, where you see the two adult birds are our surrogate hosts. Both of those hosts are layer genetics, so they produce a lot of eggs, and they are bred for the production of eggs for sale in your marketplace. And you can see the chicks standing next to those parents, and they look very different. They have dark skin, and they actually have dark meat. They have black skin, black meat, but they have white feathers. So they look like a broiler, but they're very different genetics. So those two adults, while they have the genetics of a layer, the germ cells in those chickens are actually from this breed called a silkie. It's actually a broilerized silkie bird. So when they mate and they produce eggs, the only thing that will come out of those birds are the silkie breed. And so that's just showing you that we are investing in the technology itself, gene editing technology, to improve disease resistance, but we're also investing in the technology to create tools in order for us to fully utilize the gene editing technology. And thank you.
the final presentation before we get to questions and answers. And I see in the chat box, there's already some questions coming in. Please do think of questions you want to ask this panel. Our final speaker this evening is Apollinaire Duquesne, who is the director of the Bill and Melinda Gates DFID-funded Centre for Tropical Livestock Genetics and Health, which has a base both in Edinburgh, but also in Nairobi at the International Livestock Research Institute. And Apollinaire is going to tell us briefly in the next few minutes about his views of this technology within that part of Africa and further afield in the low and middle income countries around our world. Apollinaire.
Thank you, Bruce. Hello, everyone. It's really a pleasure to offer a perspective on genome editing, but I'll focus on the potential and application of this technology in Africa primarily. And I think the same principle can be used for other low and middle income countries. Previous speakers, they've talked about the diversity, they've talked about the species and stuff like that. So the population and the pressure is really on our food systems. And I think we need a step change. Let me start by examining animal agriculture in Africa. So the challenges are what we see on the screen here, or some of the key challenges are what we see on the screen here. Productivity and welfare are critical. And currently, if you take a dairy cow, the average production is really like 1/10 of what in Africa compared to what you'll see in Europe. And welfare is an area where we have to do more. Environmental impact, we need to produce sustainably, we have to reduce the footprint of livestock on the environment. And profitability is also another challenge because we're talking about systems where production is really in the hands of smallholder farmers. What can we do to incentivize them to be running profitable businesses? So with that, let me refer you to the diagram that we have here. I just want to draw your attention to a few points. This is a comparison of the demand of livestock products between 2005 and 2030. And we see here that sub-Saharan Africa is really circled in the dark. And then we see here that at least we have to be doubling production, between now and 2030. Are we on course to do that? I think the simple answer is really we will struggle to get there. And for all of us here, regardless of where we sit, we've been affected in some ways by the global pandemic of coronavirus. And during this time, we've seen that our food systems are really weak. If they're weak in advanced economies, they will be even weaker in low and middle income countries. Therefore, we need a transformation, we need a step change. What I'm trying to convince you here is that genome editing has a role to play, and in fact, a major role to play for that transformation. So what's the landscape like in Africa? The landscape in Africa, is really, it's very interesting. It's not a foreign concept. You'll see here on the right, this is about 2 years old slide where we have about 15, 20 countries where gene editing is being talked about. It's being talked about from the research angle, from policy, regulation, and how to communicate it. And we are doing this building on 20 years of GM in Africa, and learning on how to do this. And I think the next thing I want to tell you on this slide is that the landscape has both crops improvement and also livestock improvement. And we see here in crops that we have national players, we have sub-regional organizations, and we also have international organizations within the CGIAR, and a good size of industry partners are also playing that role within sub-Saharan Africa. The next thing I want to talk about is really around livestock. We're looking at different species, cattle, poultry, and small ruminants. And Rachel talked about, the application in poultry. And I want to argue here that what she mentioned in advanced economies will be very relevant in low and middle income countries. And Anna also talked about fish. And I think the aquaculture is a growing industry in low and middle income countries. It's my personal bias that I did not add that here, but the aquaculture is also very important. Finally, there is significant number of players in terms of how do we communicate this, how do we influence and educate and bring awareness in terms of good policies for good regulation from a good understanding of what this technology can offer. So beyond the landscape, I think I want to argue here that for those who know the livestock and the animal science in sub-Saharan Africa, in the tropical parts of the world, is that we are benefiting, we are sitting on a very rich biodiversity, and we can really harness that biodiversity. And there are good examples in cattle gene editing, genome editing, the tolerance to heat based on the SLIC gene is a naturally occurring mutation that can now be used to accelerate introgression of that trait in different genotypes. And we also have the pole gene reducing or eliminating the horn, again, a very naturally occurring mutation that we can use to accelerate. And I would also argue that you can have very similar traits in a range of species going from adaptation, productivity, and all other traits. The next thing I want to say is our own center, as introduced by Bruce, is really very active in this area, both in terms of the work that we do in our UK nodes and also in the Africa nodes. And we are currently focusing on really harnessing that biodiversity to understand natural variation to improve performance. We are also linking that to other assisted reproductive technology to ensure that whatever germplasm are improved, they can end up in the hands of smallholder farmers. And finally, it is important that we control, we understand biodiversity, and we also conserve it. Let me finish here by thanking, by acknowledging the work that is being done by a number of colleagues here in Nairobi, Steve, Kem, Christian, in Edinburgh, Mike McGrew, and Simon Lilico, and finally Margaret, who is a real advocate in the landscape in Africa, and leading the communication piece for policymakers and other politicians to make decisions based on a good understanding of this technology. Let me also acknowledge the funders of the work that we do, from the Bill and Melinda Gates Foundation, the UK government through DFID, and also BBSRC. And finally, many of the organizations that are leading this work in sub-Saharan Africa and other low and middle income countries more broadly. Thank you so much for your attention.
Thank you to all the panelists for that brief presentations of different parts. I hope the audience realizes there's a little bit of hardcore science in there. There was a description of different types of applications, both of terrestrial and aqua and airborne animals, if indeed poultry do and chickens do fly, which they don't do very much. And also the different perception from industry and from Apollinaire to do with the low and middle income countries and the needs. I see there are already starting to be some questions in the chat, and I would welcome any more. But before we go there, I would just like to start the discussion by asking a question to Rachel first, and that is, Rachel, how will the consumer benefit from the application of genome editing technology in livestock protein production?
So the consumer will benefit in multiple ways. First of all, the animals we're improving the health and welfare of all of our animals, which means those birds are, when I say animals, I really mean birds. So those birds will be available for the production, protein production, and therefore be available in the supermarket. So it will be access to affordable protein globally. Secondly, during outbreaks, especially of avian influenza, exports cease. So there are very strict rules on the export or non-export of any animals that are infected or have been near within a geographic radius of any avian influenza positive flocks. So that also stops the production of chickens and the supply of chickens globally.
Thanks very much, Rachel. I will come to the questions in the chat first, but I'm going to go specifically back to Apollinaire and ask a different type of question just to get people thinking. Apollinaire, we present this from the need of feeding people. We present this from this technology from the perspective of animal welfare and preventing disease and the pain and distress that disease causes both on the animal but also on the handler and the owner of that animal. What is the perception, in your opinion, what is the perception in Africa at the minute, in Kenya and other parts of East and Central Africa of this technology? Is the African farmer keen to see application of it, or is it still something which is seen as a rather remote possibility?
It is work in progress. I think we've learned in the African scene to flip the narrative and be able to speak from the farmer's perspective. I think farmers are beginning to see that we are talking about resilient animals. We are talking about animals that will not die. We are talking about animals that will produce, and then they will make their operation profitable. So I think there's a lot of progress there. And that comes from the awareness which is really being talked about. The other dimension also is that, I talked about this work being done in Africa. It's no longer seen as a foreign concept. It's being seen as some work being done within Africa and with African scientists also involved in it. I think that has also, strengthened the argument and also strengthened the knowledge. And it's all about trust. I think overall the trust is being built.
Thank you, Bona. So I'll now turn to questions from the chat. And this is for you, Anna. It's a very specific question. Could you go back over why, the question is, why do we need salmon that are sterile? You're still on mute, Anna.
Sorry. No, the primary reason is from environmental reasons, because all the farming is done in sea cages of salmon. Most of their life cycle is in sea cages, and then they can escape, and they can also then introgress into the wild populations. And we have about 200 rivers, salmon rivers in Norway, and there's very few of those wild salmon in comparison to the wild salmon, so compared to the farm salmon. So we don't want them to genetically introgress into the wild population. And secondly, when the fish can also come into early maturation in the farm, and then the welfare of the fish is very bad. They stop growing. They also get disease. So the robustness of the fish is not very good when they enter maturity. So you don't want to have that in the farm. And the third reason is that the producers also want to protect their brand. They want to be sure that what they sell is only theirs, is IPR. So that's the third. But that's not the major issue. The major issue is the escapees.
Maybe I could ask an extension to that, and is the technology and approach that you're taking in salmon, do you believe it's applicable to other aquatic species?
Yes, I think so. So we are not working with any other species, but we know it's applicable, I guess, in model species, but also now we're working with a group working on sturgeon. where it can probably be applicable. So it's not only salmon.
So again, for the audience, the approach of creating infertility in animals is also used in the more traditional livestock that people would understand: cattle, pigs, and ruminants, and also in poultry, for a subtly different uses, which range from allowing precise genetics to be distributed within a given geographical area or a given farming environment, but also potentially having application in conservation where these animals could be used as a surrogate host or a surrogate sire to allow animals which are under some sort of environmental pressure to be sustained. This is very much experimental work, which is still in the lab, but as described, the technology works from the point of view that the animals can be, in the case of pigs and cattle, we can get male infertility into those animals. And now it's the use of that and the application of that which has been looked at by both the academic and the commercial sort of arena. Maybe I could extend the question. I asked Rachel what was the use of this technology, or what is the benefit of this technology for the consumer. And I know Apollinaire, the farming environment in the Western world is very often different from the farming environment in Africa. But could you maybe say, Apollinaire, maybe you could say a few words about what would be the major impact on the farmer? Is it something to do with the economics of the smallholder farmer, or is it much more pervasive than that? In that having animals resistant to disease will allow a much more stable farming and family environment.
I think what we're learning is that smallholder farmers are now considering options to continue to keep animals or not. We know that a good number or proportion of smallholder farmers do it from a cultural perspective and stuff like that. But now I think we're increasingly seeing, I want to call it an industry where profit is becoming an important dimension. So I think that's really, in terms of priority, a top thing that smallholder farmers would like to see, like to have access to germplasm with the traits or the characteristics that are immediately addressing their issues. Egg production, milk production, and growth, and resilience and survivability are very important key traits there. I think profit is now emerging because the demand is now quite strong. And one of the things that I would like to say is that, in terms of livestock agriculture, a significant proportion of what is produced is consumed locally. So the pressure is actually locally. So I think more than, in some cases, 90% of what is produced is to consume in the local market. So, and those, that demand is very visible to farmers. And they are aspiring to meet the demand, and therefore the quality and the type of the product that they would like is very specific, and it should meet balancing their books.
Thank you, Apollinaire. There's another question in the chat, and it's to do with a slightly different topic, and that of xenotransplantation. And I think the question really is asking, can this technology be used to generate probably pigs that would have application in the medical arena of xenotransplantation? And the answer is yes, it is, and yes, it can, or yes, it can, and yes, it is, whichever way around you want to look at it. The technology certainly can. It is accelerating some of the genetic modification events that that industry are looking at. It has not, for those of you who are familiar with the xenotransplantation arena, It hasn't generated the sort of final goal of an animal which is completely, or an organ from an animal which can be transplanted into a human without immunocompromise treatment in addition to that. However, it is certainly generated animals that can produce organs that require less immunocompromised treatment of the patient. So yes, this technology can be used in that arena. If you want to consider it though in a sort of global context, if we use this technology to produce xenotransplantation animals, probably pigs, we're probably talking across the planet in the order of the range of tens of thousands at an absolute maximum. Certainly not today. Today we're talking in the tens or maybe hundreds. But if that technology was ever to be applied, then it would probably be up in that tens of thousands. If we use this technology in traditional breeding, whether that's in Western world or in low and middle income countries, we're talking in the hundreds of millions of animals. It's a completely different scale of application. And there's a second aspect to this, obviously, in that in agriculture, these animals are part of our food chain, while in biomedical, they are part of a surgical intervention to help the patient. But to answer the question, this technology certainly can and is being used in the field of xenotransplantation. It is also being used to generate what are called large animal models of disease, where a known mutation causes a human disease. This technology can be used to generate that mutation in an animal, a large animal, and that animal can then be used in accelerating the development of therapies for ourselves. And in that context, there are arguments around biology and the similarity of biology between different animals and ourselves. And there's also arguments around the scale of the physical scale of the animal, and therefore the scale of the treatment, the drug, whatever your treatment of the disease is, can be done at a scale which would then be immediately translatable into a human patient, if indeed that is where it ended up. Both the xeno work and the disease model work is very early. There are examples around the world of this technology being used in those fields. And this is leading me to a question which I'm going to ask all three other panelists. But the timescale for how that would be applied is quite far off. And the question I would like to ask the panelists is, we've described between us different examples of how this technology can be used. Apollinaire describing, for example, the SLIC mutation that confers heat tolerance to ruminants. Anna talking about the sterility trait. I mentioned the PERS, and Rachel described a few of the examples that Cobb Van Tress are looking at. I will start with Anna, and then go to Rachel, and then go to Apollinaire. How long do you think before a genome-edited animal will come on to regulation and aspects of that in a minute? But if there was clear regulatory pathway and a clear consumer demand for it, how long do you think it would take for the current projects to reach the dinner plate? I'll start with Anna, and I'll go to Rachel, and then Apollinaire.
I guess it depends on where you are in the world, and what demand it is, and certainly which trait you have. I'm not certain that the sterility trait will be of such demand, although it's a very good environmental solution, I think. But if we manage to make a disease-resistant trait, that we just move like a wild one or something that's in not a big change, and it will be a very big benefit for both the surrounding fish and for the fish in the farm. I don't think that it's going to take very long, even in Norway or any other place where they do aquaculture, because the fish is usually not considered, at least it's not so close to us as humans. So I think maybe it will be not that long if we have a really good, desirable product.
So you mentioned, before I go on to the other panelists, you sort of differentiated there between the sterility and disease resistance, and I think there are perceptions. I mean, aquaculture, the predictions by many is that the largest growth in productivity, or the largest product, should I say, would be in aquaculture. It's going to be difficult for animals that require land-based farms or pens or sheds to dramatically increase, since much of the planet is already, that can be used for that, is already used for that. But aquaculture in our oceans, in our rivers and our seas, the perception that we can see really dramatic increases in the scale of aquaculture around the world. And obviously, exactly how big it will grow depends on which survey and which report you read, but they're all the consensus is there's going to be a massive growth. With that massive growth, do you think the sterility trait, or the ability to give that differentiator, either from a commercial or from an escape perspective, will become more important?
Yes, I do. I think so, because obviously this will not disturb the wild populations when they escape, which is important. And then you can put the farm wherever you like. Also in areas maybe where they don't have this species of fish, for example, you can put them. So I think if you can make it like in sort of an easy protocol to do this, I think it will be a desirable trait. And also they have, with the climate change, increasing temperatures in the sea, definitely you get more maturation in all farm species. So that's also a big problem, and this will be prevented if you have sterile fish. So that's a big welfare problem with climate change. So it sort of comes together.
So it is possible then that some applications of this technology can actually be used proactively as we see aquaculture grow dramatically. I think that's quite an interesting aspect to this, because in poultry and livestock, we are reacting to what's on the ground already, and the differential scale that aquaculture will see over the coming decade, I think opens up additional opportunities. Rachel, I'll go to you and ask the same question.
So I think the answer for poultry is it's going to take many years at this point. So this is not a product that you would see tomorrow. And just before I go on, I'd like to emphasize the point that this is all in research and development. None of the work that we've done in gene editing is part of any production or product program at the moment. This is all just R&D for us. So if we had a direct route and regulations were clear and quick, so getting regulatory approval was very clear and quick. If we have done all the validation of what does this gene edit do for the product, and does it have any other implications? Does it have any other impact on performance of an animal? So let's say in a perfect world scenario, we could produce a chicken that was resistant to avian influenza. It also performed well, so it was very feed efficient. It had very good growth rates. It had very good other welfare and health characteristics. So in a perfect world, we had the perfect chicken. It would still take at least 5 years for us to get it anywhere near a consumer level. And the reason for that is numbers. When you look at the poultry industry, we do all of our genetic improvement up at the very pinnacle of what we call a performance pyramid or a production pyramid, where we do all of our genetic improvements or our improvements in our product at a pure line level. But the product that we sell and the product that you eat is not a pure line, it's a crossbred. So we have to be able to produce enough numbers down our production pipeline so that we can make the crosses that we need and provide those to our customers. Our customers then make another cross and produce the product that you see on the supermarket shelves. So it is at least a 5-year process if it was a perfect world and we had everything sorted out. Now, we're not there yet. We haven't finished the R&D. We don't have a clear regulatory pathway. So it's very difficult at this point to say.
Thank you, Rachel. I will pass on to Apollinaire with the two views there, one from industry, I suppose, and one from academia, and saying very much the same thing. And I will chip in my tuppence worth at this point. I think seeing true application within 5 years is very unlikely. I would be extremely surprised if we don't see application within 10 years. And it's that window where we'll see it. And it's a combination of where projects are within the research pipeline, the experimental pipeline, but a combination of that with regulatory aspects, which I think we'll come on to in a minute. So I go to you, Apollinaire. Given that's the timeline that we're proposing is realistic, do you see this being deployed in that timeline in Africa, in low and middle income countries, or do you see additional hurdles that will need to be addressed?
I think that I would like to approach this from stating what are the bottlenecks. I think one major bottleneck is really around the regulation and policy. We all agree on that. But the good news is that there are at least 4 countries in Africa where, heavy discussions are ongoing. But, the timeline is not something that I can state. So the regulatory environment is being improved, and I think things are moving, good communication and good awareness. I think from there, there are countries that, are taking the lead on that. But the lead also comes from what's happening from the crop world. In the crops world, there is quite a lot of progress. And some of the, then the second bottleneck will be, what are the products that are available? From the crops world, there are a number of products, and some of them are actually in what the plant breeders call confined field trials. They've gone through that. They've been demonstrated in different countries, and that's going through all the safety tests and the rest of it. So that's very good movement. But on the animal, on the livestock side, there are also good products that are available. The pole genome edited cattle, for instance, and the slick genome edited cattle. These are prime products for the production systems that we are talking about. So I think the first bottleneck really pushes us in a way that, I agree with you, a 5-10 years horizon is what I will be very optimistic about, to get these policies to go through it. But in parallel, in terms of product availability, there is quite a bit of movement, both in crops and also in the livestock side. But for the livestock side, because we do have some of the elite products, I would call them, elite product, the slick and the pole genotypes that are ready to go, essentially. And one final thing that I should mention here is that it's not the best option, but traditionally in many production systems in Africa, germplasm have been imported for crossbreeding. And I would see that if the landscape has a green light, we could have very rapid movement. But once again, 5 to 10 years would be, the horizon that I'll be looking at. But also the pressure is mounting. COVID has demonstrated that our food systems are very vulnerable, and then doubling or more than doubling production is really important. And the horizon 2030 is also, putting us in a lot of pressure. I think all those things combined can maybe at best, reduce the 5 to 10 years horizon for the first products to be out there in utilization.
Thanks very much, Apollinaire. So again, for the audience, around our planet, different countries, different environments have progressed through the regulatory world at different rates. And there are countries which today have legislation which you could take a genome edited animal through and get and some individuals have taken these animals through to a point where they are allowed to be used and be put into an environment. They haven't been done yet. There's not actually such an animal through there, but there are regulatory differences that exist around the world. We often look to the USA, to the FDA and the process that they're embarked with. I'm sure many of the audience are familiar with where we are within Europe and our debate around the whole space of genetic engineering and aspects of it, and subtle differences between the regulation between what is a product-based one and what is a process-based regulation. We could spend an awful long time trying to delve into that, and I don't want to do that, but I want to ask each of the speakers their opinion about harmonization. How important is it for this technology that we see harmonization of regulatory positions around the world? Or is a situation we currently have where some countries would allow it and other countries are yet to either decide, or in some cases do not allow it to be used, will be, if it becomes pervasive, it will be a successful and manageable system to work with. So I will jump straight to Rachel, because probably this is of most interest in the commercial arena, and then I'll go Anna, and then again I'll bring Apollinaire in. Rachel.
Yeah, so harmonization is one of the most important factors that we have to consider in order to distribute products globally. When you look at the company that I work for, Cobventris, we distribute to over 100 countries globally. And then those countries distribute it to other countries. So if we don't have harmonization, we will be in a state where we can supply this product to some areas, potentially, but not others. It would really, without harmonization, we would really be hamstrung in our ability to distribute this product. Now, that's not for saying that, you know, when we are through with the development of this product, and if we had regulatory approval and we were able to distribute this product, we would end up distributing a product that is non-gene edited, of course, and gene edited, so that the consumers still have a choice. But you still need harmonization, you still need agreement across countries in order to ship our product globally.
Thanks very much. I'm going to actually change my order. I'm going to go to Apollinaire because I want to ask Anna a subtly different question here. But Apollinaire, what is your feeling about the need for harmonization of the regulatory environment around genome editing technology?
Yeah, harmonization is critically important, especially in Africa, because you need trade across countries, and one of the challenges that we face in Africa is that trade across countries is not enough, and therefore economies suffer. So for this to be successful, you know, harmonization either at the Pan-African level or at least sub-regional level is important. And we have sub-regional organizations that are already set up and they're talking to each other. They have economic regions. I think the system is perfect for it. Without harmonization, things will not move as much as we would prefer. So I think for me, it's a critical piece for this.
So Anna, the subtle variation I'm going to ask you, it's about academic effort, academic location around the world. I would say that Europe has been very strong for a long time in genetics in general, but also embracing the technology around genetic engineering and now genome editing. It's not alone. There's an awful lot of effort going on around else in the world, other places in the world, but Europe does have a good strength of academic effort in this technology. Do you think that a lack of harmonization within the regulatory and perhaps within the funding arena will lead to migration of the scientific, the so-called brain drain out of some parts? Would a very beneficial regulatory environment in some more southerly clime entice you away from Norway, for example?
Well, no, I hope not, because I mean, I think I'm interested in you know, I'm a little bit interested in relation to the regulation. I'm a little bit more interested in the application. You know, whether we can produce something that's environmentally friendly, that's good for the communities, and that's another type than saying that everything that's edited with this type and this type can be approved. It's more like what's the benefit? Can we do something that's very beneficial for society and the people who lives there? That's overcoming this risk thought that maybe it has some kind of risk, which is, if we can show that with our science, then I think it will change, because it has to be scientifically sound. And if the application is too good to be true, which I think is with this technology, I think it will come, but you need to have those demands. Maybe like those products that are around now are not like very high demand. That's why they are not required enough. But of course, Europe is very crowded. We need environmentally friendly solutions. We need to be able to produce food from all the people in a sustainable manner. So if any of those genetic changes that we can do helps this, the development of a sustainable living in Europe, which is very crowded community, I think that will come through what people want, not what regulations want.
So that's a really interesting angle. And you're probably already guessing my follow-up question, but I'll preempt it. Well, I'll say the question and then I'll talk about it a bit before I come back to you on it. How do we find out what is a societal need? How do we find out, in your words, what the people want? I know there are a number of surveys, there are events like this which we're participating in, and there are numerous of these around the world. I know that there are different surveys and different questionnaires that go out, and we get an opinion back from that. But how do we, what are your thoughts about actually engaging in a proper societal dialogue about what bits of the technology or what applications of the technology should we go after? The academic world is very much pushing at the minute disease resistance, and we've all mentioned this at length today, and we all have examples, and I presented the PRRS story, but we're working on at least another 4 or 5 other diseases that we believe, or to be more precise, we're working on viral diseases, and one of the reasons for that is the virus itself has to get into the cell to do the damage. It's not able to do damage independently. It has to get into the cell of the animal to do, and therefore there is a moment in that life cycle of the virus where it interacts with bits of the host cell membrane, the host internal proteins, and so forth, and they're all candidates for us to edit to give sort of resistance. So there isn't not to say that it's the easy option out there for the academic and experimental work to go after, but it is a real option in that we know there must be candidates for that. When we go into other traits, other diseases, bacterial diseases or protozoan diseases, we're one step removed from that interaction, in that it's the parasite or the pathogen, is interacting with the animal rather than necessarily, sometimes it's with the cell, sometimes it's intracellular, but often it's interacting with the animal rather than the specific cells. And then we go into traits that Apollinaire mentioned, the POLD, which relates to the horns of the animal, and the SLIC relating to heat tolerance. Again, very complex biology, which we're trying to go after. So you could argue that we are going after the easy end of what is a complex and very difficult biological challenge. I think the evidence from POLD, from SLIC, and some other projects indicate that we can go the next step. But if you look across the board, the majority of projects are in disease resistance, and the majority of those at the minute are to do with viruses. So academia, in conjunction with industry in some places, but definitely academia, our universities and our research institutes around the world have picked on disease resistance. This is the first one. It hasn't gone out to society and said, is this important? We know today that disease is important. It's transforming our world. There's a certain virus which is going around our planet doing an awful. We know that disease is very important. We know that disease is a major burden in farming, livestock, poultry, aquaculture. but we haven't gone out and asked society about this. You know, we as academics, how do we engage with society? How do we find out?
Well, we have been
Areas.
out asking society. We have been out asking people around the shopping mall and around to ask them what they think. Do you hear me?
Yes.
Yes.
And it's been very interesting, and I recommend all the scientists to do this, because I mean, we usually don't do this. So we had people helping us to do this, because you get this reflection into the lab. It's quite interesting. So then you start to believe when you hear, do you hear me?
Yes.
I had this very nice talk, and there was this, you know, that always this young nature organizations, they're against everything. And they approached me, one of those organizations, said, but your solution is good. How can that be? Because you're changing genes, you know. So it's sort of, you have to talk to people. We need to meet people. As scientists, we cannot only sit and think about the mechanism, although that's what we want to. But if we're going to, we have to engage in society.
I think we also have to. Sorry, I'll interrupt. I think we also have to engage in more than just asking, you know, where do they think this is applicable? I think we need to be engaging in certainly in the younger generations, but all generations, is what is available, what opportunities are out there, what are the problems? Because I think in today's society that so many people are distant from the production of their food. And they don't really, well, I'd say there are many groups that don't really understand the challenges that we have to face. And why do we need these solutions? So I think it's also not just an engagement, like Anna was mentioning, but it's also an engagement with society, with groups in society, showing what is the problem, what are the challenges, what are some potential solutions, so that they can make an informed choice.
Exactly. So I think that might be easier in smaller countries than larger, because you have some, I don't know, but we just have to include that in our daily tasks as scientists. I think that's important.
So I know Anna didn't say it. You could see in how she responded to the question that you actually get enjoyment out of this dialogue with people and opening up questions. And I would agree entirely that as academics, we speak sometimes our own language, and we certainly discuss our science at great depth with colleagues. But it is very, very enjoyable to broaden it to non-scientific, to understand how it's perceived, because as everybody, we live in our own worlds, and obviously we're comfortable with those worlds. And I would say enjoyable type of way to go out there. I'm realizing we're running out of time for our slot here, so I'm going to ask each panelist in the order of Rachel, Anna, and Apollinaire to just summarize their feeling about this technology, where it's going, whichever bit you want, but just a couple of sentences, a couple of phrases to indicate where you think we are, or why it's useful, whatever aspect you want. Rachel.
Sure. I think that gene editing has great potential to solve some really big global issues for health and welfare in livestock species, and I throw chickens into livestock. I think there are many challenges for food sustainability, and we really need to start tackling those with technologies that are able to answer the questions, and that will be transformational changes, not necessarily just incremental changes.
Thank you. Pass to Anna.
It's a difficult question, but I think, I mean, if applied in the right way, if it helps society, I think it will come into use not so long from now. It depends on the product. And I think also we'd have to think about this technology as helping us understand the biology a lot. It's not only the application, it's a lot. It's going to take us forward in so many ways that it's not going to lead to edited animal in in production, but maybe we can make better vaccines, we can make better treatment, better things in all ways, just using this technology. We see that a lot now with the science we're doing. But if we get the application that the people want, I think it will come into use not so long from now.
Thank you very much, Anna. And I'll pass to Apollinaire.
So I agree entirely with what Anna just mentioned. Genome editing is a technology really to help us generate some products, but it's really a good tool for functional genomics. It can help us to really advance our understanding of important biological questions. And I think I would like to always maintain the two dimension. But in terms of applications, we touch on, what are the traits, what does society think about and stuff like that. I think we're beginning to see that, at the dimension, the socioeconomics, the social science dimension in contextualizing the sort of things that we believe could be an intervention. I think we should do more of that. A product that is disease resistant or heat tolerant is a good thing only when it fits within a context that is going to be accepted by society. And I think that always, analyzing that in the context of food systems, then we're going to be clear. The scientific community, the rest of society, we agree on what the challenges are. If we agree on what the challenges are, then there is expectation that we will agree on what the tools or interventions would be. So those are my two last comments.
Thank you very much. I'm going to say that my simple phrase, and I'm stealing some words from others, is that the genie is out the bottle. This technology is here. It's not going to go away. It's up to us as individuals across our society, academics, researchers, industry, consumers, suppliers, farmers, everybody to work out what is the good applications. I would be very strongly of the opinion that some of these early ones, certainly some of the disease ones, have utility, and we need to explore how they could be used, and that will help us understand how to use this technology going forward. I see there is a little question from the audience here, which I'll just pick up. While I do that, Ines, are we still able to put the question poll website back up again? When talking about, this is the question from the audience, when talking about genetic engineering or genome editing, we talk about consumers. I've lost it there. Could we, I'll come back to that question in a minute. Could we, again, oh, there we are, thank you very much. We talk about consumers' concerns, regulatory problems, but what about farmers? Is there a difference between African and EU farmers using edited animals, genome products or offspring in their farms? And a very good final public dialogue is the heart of ISOF and a good place, and that is exactly why we're here. So maybe I could ask Apollinaire just to touch on that very briefly, and I think it's probably more important to touch on the question than do the poll actually. Do you believe there's a big difference between the farmer in Africa to Europe with regards to wanting to use this technology?
I don't think so. There is no fundamental difference. But we also have to acknowledge that these two types of farmers are facing somewhat different challenges. But their targets, they want to run profitable businesses. They want a product here being an animal or a bird that will fit in their production system. So I think the perspective is really, they share the same perspective, but the challenges may be somewhat different in terms of what are the key challenges and specific, what disease, what production systems they're operating in and stuff like that. So that's, yeah.
Thank you very much. I'll just add to that to say that there's obviously elements of scale difference between farming in many parts of the country, many parts of the world, and as Apollinaire alluded to, there's traits and different priorities. But at the heart of it, a farmer wants to produce food for sale, for his family, for his livelihood, and wants to do that in the most efficient way. And for the vast majority of our farmers, the vast majority of farmers want to do that in good welfare conditions. If we only have a couple of minutes left, and I think going to the vote might be just a little bit too tight, which I apologize if people have been waiting for that, but I would just like to thank all the panelists. That's Anna Borgelius from Norway, and long may you stay in that beautiful country. Rachel Hawken from Arkansas, quite far away, and thank you for joining slightly early in the day than the rest of us. And of course, Apollinaire for input across a different arena to what we normally encounter within Europe, and myself and colleagues from Roslin. Ines, I'd like to thank Ines for all the background and help for this, but also I'd like to thank ASOF for the opportunity, but also for the technical input. And at this point, again, I'll apologize for certainly my disappearance earlier on in the session. It is amazing that we rely on technology, and sometimes it just doesn't quite work. I would like to finish off by saying the blue skies that you see behind Apollinaire are exactly the same as the blue skies I see outside my window as evening starts to come in Scotland. And I hope that weather, or nice weather, fits you all around wherever you are. And I thank the audience for their participation and questions. Once again, thank you to the panelists. And at that, I think we'll finish this session and look forward to opportunities where perhaps we could do another session in the future, but face-to-face, which is always much more appealing and enjoyable. As Anna said, speaking to people is what's nice on this planet. I wish you all well, and thank you for your input. And at that, I think we'll finish the session.